Hello, everybody, and a warm welcome to our audience. I see most of you are logged on already. I welcome you to the second part of the SICAM conference 2021, although it is already 2022. I wish you a very happy new year. We haven't seen each other for a few weeks, time to celebrate with our loved ones. I don't know how it is in your countries, but in Switzerland, we have had the warmest New Year Eve ever since recorded history. So I think, well, Alice is nodding, so I guess it's not, Switzerland hasn't been the only place, although it has snowed again in the meantime, but it is very spring-like and very unusual. So I think that will occupy us a lot in the future, this kind of climate. So tonight is a special day. We have two talks tonight to present to you from the lab, from Alice, from the Mountain Research Center in Bragança in Portugal. I'm very happy that Alice is also here and able to host tonight's talk. We will hear a talk from Dora Herniques. Hello, Dora. Very nice to have you here. And we'll have a talk by Carlos Yadro. Hello, Carlos.
Hi.
Nice to have you too. Both working with Alice. She will talk about what they're talking about later. I think everybody's very excited. It's about morphometry, so, you know. Alice is a professor at the Polytechnical Institute of Bragança at the Mountain Research Center. She's also part of our scientific board, so part of SICAM. She's very accomplished in honeybee genomics. She's done her PhD in the US about honeybee genomics. She's also worked with Stephen Shepard, which some of you might know. She's worked on the Africanized honeybee then, I think in the southern US, and then came back to Portugal and worked on the differentiation of honeybee subspecies and evolution ever since. And she will tell you about Dora and Carlos's accomplishments later on. So tonight's structure, to inform you, we have Dora first with her talk about the color of, the differentiation of the color among honeybee subspecies. Yeah, very pressing issue. We will have time for a talk and following questions about 45 minutes. Then Carlos will have his talk, also with the Q&A following. So we will have to cut Dora's Q&A at some point, but if you have pressing questions, we can still add them after Carlos' talk in the Q&A session then. So don't worry. Okay. So I will hand over to Alice and wish you all a very pleasant and interesting evening.
Thank you, Gabrielle, for this introduction. And indeed, we had a very warm New Year's Eve, 21 degrees. I went to the beach, very unusual. I also want to wish everybody a happy New Year and a healthy New Year, and I hope that the next conference of SICAM is alive. This is a very nice format, but I miss seeing the people. It is a pleasure for me to host the first session of the year, and in this session we will have two talks. The first one is going to be done by Dora Herniques. Dora was introduced to honeybees a long time ago, about 10 years ago, when she came to my lab to work on a project that dealing with the genetics of Iberian honeybees. She did a master's degree in genetics and evolution, looking at humans in Africa, in the genetics of the population of humans in Africa. So she changed from humans to honeybees, and she never left honeybees ever since. They are really catchy, aren't they? So she started working in my lab in this project. Then she got the PhD scholarship from the Portuguese National, for the Portuguese Science Foundation under my supervision. And for her PhD, she continued to work with the honeybees. That's what we do. And using whole genomes to ask different questions about genetic patterns of Iberian honeybees, looking at selection signatures specifically. And she also developed assays for introgression, SNP assays for introgression in the M lineage bees for Iberiensis and for Apis mellifera mellifera. These assays have been used by several colleagues across Western Europe to identify introgressed colonies. And actually, Dylan already presented some results of this research using this SNP panel. Dora graduated in 2018 and has since been working in my lab as a postdoc, and I'm very happy to have her. She has now a family in Bragança. She's the mother of one daughter, and I hope she continues to stay with me. I hope so, and she will. Today, she will present the results of a study on the abdominal color patterns using Apis mellifera iberiensis and mellifera mellifera as the models. And she is relating the color patterns with the introgression estimated from nuclear SNP markers. Well, she has been publishing a lot of papers, as you probably know. She has been very productive. And with this, I give the stage to Dora.
Yeah, thank you very much. I will share my screen. Can you see it well? Good evening, everyone. Thank you very much, Alice, for your nice presentation about me. You always have, you always talk very nice things about me, and I don't have words, you know. When I did my PhD defense was the same thing. I cannot talk about it. Thank you, Alice, and I really enjoyed to work with you, and you know, we hope to continue work with bees and continue try to protect them. So thanks also for this opportunity to show one of the works that we have been developing. This one particularly, Can We Tell an M Lineage Honeybee by its Color, was a collaboration between Alice Groups and Grace from Ireland. Let me just, I have a small problem here. Okay. So we have 31 subspecies, and this is the natural distribution of the 31 subspecies. And all this diversity have been grouped into 4 main lineages. And of course, in the same lineage, we have the subspecies that are more closely related to each other. So we have the Eastern European lineage, or C lineage that contains the subspecies that the beekeepers prefer. We have the O lineage, or Middle Eastern, African, or A, or and M, or Eastern European. And now we also have in this lineage the new shiny subspecies that I cannot tell the name. Now we study this diversity using molecular markers, but before this diversity was studied using different characters, and this is just a table that I took from Huttner's book with an example of different characters that can be used to study this diversity. And we can see here that the color can also be used. Actually, color was one of the first characters employed in the early honeybee systematics. However, honeybee is not used in academic works anymore, at least alone, because it's not suited for discriminant analysis. because it is a bimodal distribution among populations. However, we cannot forget that it is very easy to classify in the field. The trait that is easy to classify in the field, and because of that, the beekeepers like to use the color a lot. There are some old studies that try to understand the genetics behind the color, and they found that there are several genes related with the color between 3 and 7, and also that the irritability is around 0.3. This means that the genetics is important, but the environment is also important. In Europe, we have two subspecies of two lineages. We have the M lineage, the Apis mellifera iberiensis, that is in Iberian Peninsula, and Apis mellifera mellifera. We can see the distribution, the wide distribution here. And we also have the C lineage that contains 8 subspecies and has Apis mellifera ligustica, our yellow bee. We all know that there are introductions of C lineage queens, and this has been changing the natural distribution, mainly of Apis mellifera mellifera. Some local populations disappear or almost disappear. The story of Apis mellifera iberiensis is a little bit different. Several genetic studies show that Apis mellifera iberiensis is well preserved. But this is for the native range. The story is a little bit different for Canaries and Azores. Now the people understand the importance of the conservation of local populations, and because of that, the beekeepers try to keep certain types of bees. For instance, in Ireland, that the beekeepers want to keep the black bee, Apis mellifera mellifera, they try to select just the black bees, so they eliminate the bees that have the yellow color. On the other end, Azores try to keep yellow banding phenotypes. We cannot forget that there was no bees in Azores. They were taken by the Portuguese settlers in the 15th century. And after, in the 1980, there was a program and a lot of C lineage subspecies were introduced in Azores. So we expect, and we already saw that there is a lot of integration in Azores. But it's not important if you want to keep the black or the yellow bee, if we don't know if the color and the genetic background are associated. And this is our question. Can we use color to select the desired genetic background? To answer this question, we can use molecular markers. And in this case, there are several tools based on SNPs, for instance, and I will talk the tools based on SNPs that are single nucleotide polymorphisms, that are mutations. And that happens, for instance, here we have a G and now it is an A, and this is a SNP. And for honeybees, there are several tools that were developed, for instance, for mellifera, to calculate or to see integration between Apis mellifera mellifera and C lineage subspecies, and also for iberiensis to calculate integration for Apis mellifera iberiensis and C lineage subspecies. We can see all the steps of the development of these tools in these papers, and these were the tools that we used to associate the color and the genetic background of our bees. So we want to verify if there is an association between abdominal color, pattern, and integration level inferred from SNPs, because we can have black bees that they are unmixed. And if we keep them, we are actually promoting the genetic pollution of the population. And we can have yellow bees that are pure subspecies. And when we are eliminating them from the populations, we are actually losing diversity that is very important for our populations. This information was already published in 2020 in this paper, so I will show just some information that is here. So if you are interested in reading these and see more details, you can go to our paper. To answer our question, we genotype and we identify into 4 phenotypes, 98 Apis mellifera iberiensis that are from 84 colonies. And here in blue, you can see the distribution So we have bees from Portugal, Spain, mainland, and also from Azores and Madeira. We also use 162 Apis mellifera mellifera individuals from 48 colonies. Because we have bees from the same colony, we did two different analyses. One that we called intercolonial analysis, where we analyze the colonies from different, where we analyze bees from different colonies, where we compare the bees from different colonies, and intracolonial analysis, where we compare the bees from the same colony. So we divide all these individuals. We classified all these individuals into 4 phenotypes. We have the BLK, that stands for black, when the individual is completely black. We have the SPT, that stands for spot, and we have 2 yellow spots on the opposite side of the target 2. And after we have the B1T, that is when we have 1 band in the target 2, and in B2T that we have at least 2 bands in the target 1 and 2, and when we have more than 2 in the target 3. And it was impressive that we could see all the 4 phenotypes in both subspecies. So now I will show you the results of the intercolonial analysis. So we use individuals from different colonies, 84 Apis mellifera iberiensis and 48 Apis mellifera mellifera. And this is the graphic that everyone shows when they perform admixture analysis. Here, one line represents one individual, and the colors represent the different populations. In the graphic A, we have in blue Apis mellifera iberiensis, and in the B, we have in blue Apis mellifera mellifera, and in orange, we have the cell lineage subspecies. What we can see here is that most of, and after we have the proportion, we can see that we have different degrees of orange. This shows the integration proportions, also called key values, of the bees, of the individuals. We can see that most of our individuals, of Apis mellifera iberiensis individuals, are quite blue. This means that they are quite pure. Actually, 15% of the individuals have a key value higher than 5%, and all of them were from Azores. In Apis mellifera mellifera, we have higher levels of integration. 44% of the individuals have a key value, integration level, higher than 5%. And most of them are from the UK. I think that I forgot to say that we have individuals from the UK and also from Ireland. And most of the individuals that we see that are highly integrated are from the UK. When we try to see how it is the distribution of the integration values or key values among the 4 phenotypes for Apis mellifera iberiensis and Apis mellifera mellifera, we can see that we have a wide range of distribution of key values. In Apis mellifera mellifera, we have from 0 until 0.2, and this goes from 0 until 1. And in Apis mellifera mellifera, it goes from 0 until 0.6. What we can see here in Iberiensis is that the distribution is not very different in the 4 phenotypes. Actually, if you look to the mean, they are quite similar. And we can see that the highest proportion integration level is found actually in the black bee. But a different story, it is in Apis mellifera mellifera. We can see that we have, on average, a higher integration level for the bees with 1 band or 2 bands or 2 yellow bands. Actually, this difference is significantly higher compared with the black bees or the bees with the spot. This means that when we have yellow Apis mellifera mellifera, we have higher probability of being integrated. But I would like to show you again some results. These dots represent the integration level of the individual. And we can see, for instance, when we have 2 bands, we have also pure individuals. So if we have a population and we decide to discard this individual, we are actually discarding probably important individual and a pure individual. On the other side, if we want to keep this black individual that has more than 50% of its genome of cell lineage ancestry, we are actually promoting the genetic pollution. So we should use color carefully because it can be misleading. For the intercolonial analysis, we wanted to compare the individuals from the same colony. We wanted to see if you can use color as a proxy for integration level when the individuals are more similar genetically and environmentally. We also wanted to check if the colonies that have different phenotypes, so in the same colony we can find black, for instance, and bees with 2 bands, if they have like more variability in integration level than in colonies that have only 1 phenotype, and in our case, only black phenotype. For Apis mellifera, we only have 7 colonies with several individuals, and in 5 of these colonies, all the individuals were pure. So we could not do intercolonial analysis, but what we could see in the other 2 colonies that show some signs of admixture, we could see that was the black phenotype that had the highest integration level or K-value. In Apis mellifera, we had 28 colonies, 20 of them mixed. This means that they have different phenotypes in the same colony, and 8 of them only had black individuals. In the mixed colonies, we could see that black individuals were frequently, but not always, the lowest integrest, and individuals with 2 bands were frequently the highest, highly integrest. To try to answer the question if we have more variation in the mixed colonies, we have this graphic, and we can see that apparently we have more variability, but it is not significantly different. So if we have only a specific phenotype in the colony, or if we have different phenotypes in the color, we do not expect less or higher variability in that colonies. But where the yellow color comes from in these populations, there are 2 hypotheses. Of course, the origin of the yellow color could be the gene flow between the M lineage subspecies and Apis mellifera ligustica, but some yellow pigmentation can be also in the standing variation of our M lineage populations. And our results, and also because of that, we should use molecular tools to be sure that we are not keeping integrest individuals, or that we are eliminating individuals that could be very important for our populations. So our conclusions, our data show that for Apis mellifera, the integration was similar among the 4 phenotypes. Therefore, integration is a poor predictor of color, or color is a poor predictor of integration. In Apis mellifera, the story is a little bit different. The bees with 1 band or 2 bands exhibited higher integration values. However, sometimes we have black bees that are integrest, highly integrest, and sometimes we have bees with 2 bands that are pure. So we should be careful with the color. Concerning the intercolony data, we can see that even when the individuals are genetically and environmentally related, there was no clear association between the integration level and the color pattern. And these are the main conclusions of our work. I am supported by BIAPI, and this is my email. Thank you for your attention, and I am really happy to answer your questions. Thank you.
Thank you very much, Dora. And we have to make sure that Alice will be able to come back in again. Now everybody can pose their question, or Rachel has already posted one.
Is it my job now?
Yes, your job.
Okay, well, we have a question from Rachel Ward, and she asks whether the data suggests that bees with different banding patterns are subspecies.
No, no. Actually, in the same subspecies, we have the four phenotypes. So we cannot tell a subspecies by the banding patterns. It is not correct. It is impossible. Like at least in our data, in Mellifera and in Iberiensis, we cannot distinguish these subspecies by the banding patterns. I think that this was the question. I don't know.
Yes. Yeah. Well, there is one more question popping up from Brendan Murray. You mentioned, Dora, that most of introgression for Apis mellifera mellifera was within the UK bees. Was the Irish pattern similar to the UK or closer to the Apis mellifera iberiensis with less introgression?
Was more similar with Iberiensis with less introgression. So it's a more pure population in Ireland. Yes, we already saw different colonies in other studies, and actually Ireland is a quite pure population.
Yeah. Well, it was interesting to see that the banding patterns were really similar across the different populations coming from different geographical areas. Well, I think that in the UK, we were expecting to have more introgression because there is a longer history of introgression in the UK. But still, we cannot use color as a predictor of introgression, and this is the main message coming out of this study. Meanwhile, there are more questions. One from Vincent War. Hello, Dora. Thank you very much for the talk. It is now documented that beekeepers should be very prudent when they want to conserve local bees. Their eyes are not enough to select for native bees. Do you have evaluated the probability of mistake?
Well, here I don't have a number, but yes, there are always some yellow bees that can call yellow bees that are pure, and some black bees that are highly introgressed. But I don't have a number, to be honest. I could do that, but I don't have a number.
And now a question. Oh, it's not a question, it's just a comment from Jonathan Ellis that is appreciating the talk and acknowledging that it's good to have color studies systematically in comparison to the genetic data. And there is a question from Henk Kok. Are there bug-fast beekeepers in Portugal? Because they crossing with African bees and even queens from Turkey?
Well, we have bug-fast beekeepers in Portugal, yes. As far as I know, our beekeepers don't cross our bees with African bees. It's the only thing I know.
Yeah, there was a study that the Portuguese population is like a contact zone between the A lineage and the M lineage anyways, isn't it? But there it hasn't been like observed with the color so far, hasn't it? I think the question we all, from the practical beekeeper side, interests us, is this yellow color in the pure, is this like a long ongoing hybridization event? Like, did you have any SNPs, coding for color that are maybe like mingled into the population for so long that, it cannot be no longer be detected, or meaning that, you know, we have to deal with some sort of introgression not detectable anymore, I think.
Yeah, I understand. Actually, we don't know the genetic basis of color because the studies that have been doing that, they are really old. So our SNPs, they are not related with the color.
Yeah.
And another thing is that Iberiensis is not so closely related with the populations from North African.
Mm-hmm.
The mitochondrial says that it is African haplotypes, it's true, but when we look to the SNPs, we can see that they are quite different subspecies. But about the color, yes, it's an interesting question, because we don't know if it's standing variation or if it is something that is going on like because of gene flow.
Yeah.
But with this data and with these SNPs, it's impossible to answer that.
Yeah. So we can say at the present, in those populations, or at least in Iberiensis and in the Irish population, we cannot take color as an indicator for hybridization or not. But when there are one or two bands, we rather exclude them.
Well, we have an example with 2 bands, and the introgression proportion was 0.02, like 2%.
It's always those special individuals that.
So it is not easy to answer that like that, because even with 2 bands we have pure individuals, and this is the problem. So it's why it's difficult only using color just decide if we want to keep. Of course the people can do that and they do that, but it is not an easy decision, I think.
It's always are you excluding genetics, or are you establishing a further, you know, like going very like very narrow to have like something visual to be able to select. Like here in Switzerland, in the western part, they used to select their carnica for tongue length, which is not a natural like characteristic, but because they selected on it, they were able to distinguish their carnica from the environment. So they were able with morphometics to distinguish the false pairings until they introduced some other carnica, and then everything fell apart. So I have to be very disciplined about it. Yeah. So, but there are some many.
Yeah, just let me comment on this discussion. I think it's also important to know about the history in each region, because, for example, in the Azores, where there are many bees that have yellow color, and we can trace back to the 80s when they introduced bees from New Zealand. And since then, since Varroa was introduced in mainland Portugal, the Azores didn't have Varroa, so they forbidden the importations, and they have been isolated. But the beekeepers, they have selected, some of the beekeepers, they like yellow bees, and they have selected for yellow. And it's impressive. For example, drones, we have seen drones, they are totally yellow, like Ligustica. And this is explained by this history. But when we look at the genome and we use SNPs, we don't see a correspondence between the introgression level and the color. And this is because bees have been isolated from gene flow. And the Apis mellifera iberiensis background is going up. So they are selecting for color, so they select just one gene, but most of 1 or 2, I don't know how many genes are under are controlling the color. So if we select for color, you are just selecting a very small part of the genome, and the rest of the genome can be pure, almost pure iberiensis or mellifera. So I think it helps to know about, to make decisions, to know about the history, the contemporary history of the beekeepers. If the beekeepers are introducing bees from C lineage, maybe the yellow color, yeah, maybe you have to be careful about using, you can, I would say that maybe you can use the yellow color to select out if you want to keep the black bees. But it's also important to keep in mind that you may have black bees that are not pure mellifera.
Yeah.
I think
We had
that's,
one with 50% of C lineage ancestry and was completely black. So
That's always the thing. Like it's understandable that when you're selecting for black bees and you want to market black bees, right? You want to sell black bees. You cannot allow yellow bands because they don't look black. So, you know, it's kind of, but the other way around, it's always often beekeepers, they think, well, I select for black and then I have black, and then they don't, right? And that's where DNA analysis comes in, which is hard to accept for many, you know. So that's very interesting. I think Joe, he has a...
Yes, Joe has a question. He has used color to improve purity of his bees, and the DNA results, they are congruent with this selection. Well, in that case, good. His bees were analyzed by our group and Gabriel. Well, maybe, I don't know. So thank you. Thank you for sending your bees. And then there is a question from Adrian. He asks for if it is possible to design a genetic test based on SNPs in the exome, the coding part, because from the paper, yeah, most of the SNPs are non-coding.
Yeah, but now it's possible with all genomes that we have. It is possible to design a SNP tool using the exome. Yes, it is possible, but doesn't mean that the results will be different. Because if we wanted to study color, we should have like SNPs in the genes related with color, and we don't even know for sure which genes are related with color. So depends on the question, but yes, it is possible to design a genetic based test based on the exome. Yes.
Let me add to this, because we recently, Dora is forgetting that we published recently, a couple of months ago, a paper in Scientific Reports where we designed a SNP assay from genes related with immunity, and we tested, so they were selected to be in the immune genes, and we tested for introgression, and actually they can be used to estimate introgression genes from genomes. Some are in exons, right? And they are
they are non-synonyms, some of them.
Non-synonymous, yeah, non-synonymous, change the protein, so, and they can be used to look for introgression as well. There is a question from Rachel, and she asks, do you have a theory as to why some hives have both black bees and banded bees? We find both phenotypes in the same colony.
Yes.
So there is variation within the colony.
In both subspecies, yes. Well, I don't know what to explain.
Did you see if they have the same patry line, if they're super sisters, or, you know, with the different patry lines, if they're like with a different color, if they can be associated to different fathers, maybe?
Well, we didn't check that. They have different key values, different integration levels. So probably they have different fathers, yes.
So,
but
were they like matching, like within the same colony, the ones with bands? Did they have like the same integration levels, and the ones without bands others, or?
No, actually, for instance, when we had only black colonies, or colonies with only black bees, we also had a lot of key values, a lot of variation in the key values. And in the same for the colonies that where we have like different phenotypes.
Okay.
Yeah, we didn't do a paternity analysis because it takes a lot of bees
Yeah.
to infer the number of families or subfamilies.
Yeah.
But it could be related with the father. We just don't know. But it could also be the queen that is heterozygous and is,
yeah, it's we just know that the genotype, they are different, right? But after, we don't know if it's because of the mother or the father.
But when you say that, you know, the Azores, they selected for yellow bees and they have yellow drones, which are almost pure Iberiensis again. So how hard it is to select on color? It's probably not very hard, right? It's like with the cubital index. I remember there was one paper that they had a head, yeah, it was like a very high number. They were heredity, where they were able to select on cubital index, you know, very fast. So probably with the color, it's the same. That would be interesting, how high the heredity is of the color.
Well, there are some studies that say that is 0.3.
Okay.
it's actually high, and if it's just controlled by a few genes, it's you can make progress very quickly.
Very
And
quick.
with these irritabilities, they are quite good.
Okay.
And oh, there is a comment from Dorian Pritchard. He says that British beekeeping his friend found that when he crossed dark local bees with yellow ligustica, he obtained daughters, both dark and yellow. This is related with what we just talked about, depending on which way the cross was done. When the mother colony was yellow, the offspring were yellow, but when the mother colony was dark, the offspring were also dark. It was deduced that a nursing factor was involved, derived from the nurse bees. Have you found any evidence of this in your work?
Well,
we
didn't
don't know.
test.
We don't know, because these are bees that are provided by beekeepers. We don't know their
The cross.
the history of these colonies, unfortunately. But this is interesting. We could something that we could test. We could do some experimental crosses and try to follow.
That's always an argument that comes all over again. This maternal dominance for all kind of characteristics. Every now and again it pops up, and beekeepers say, well, also with aggressiveness, that's supposed to be maternally dominantly transferred or somehow. So that's really something that would be worthwhile exploring, I think, as well.
Yeah, definitely. Yeah.
But with the color, I find that kind of hard to believe that maybe it's a nursing factor. Like with behavior, you could imagine it, you know, makes more sense, but something physical, I don't know. Well, you never know.
Yeah, I have no idea. Another question from Lars: is Carnica bees a possible source of introgression?
Yes, can be. Well, when we did a genetic analysis, we used both ligustic and carnica, but after, we don't really know if the populations are introgressed with ligustica or carnica. We just tested like for cell lineage. But the thing is that carnica is not yellow, right? And it has a cell lineage ancestry.
Ooh, there are carnica that are supposedly, you know, also have this, it's like the same question, these little spots and bands and whatever. So carnica has no yellow. Well, depends who you're talking to.
They are closely related, so it's difficult to tell them apart genetically. I mean, we can do that with structure, but it has, after many cases, number of clusters. But in the Azores, we know that they have been importing bees of ligustica and from New Zealand. And as far as I know, in New Zealand, they I don't know. Maybe they also use carnica, but I think it's more Ligustica. I don't know. But the bees from the UK, well, they could be Carnica, Ligustica, we don't know. We just cannot tell apart the two from the number of markers that we have. A question from Jonathan. Oh, from Victoria. I'm confused because the name pops up of Jonathan and then it's from Victoria. Maybe they are together. Do you think the introgression in Apis mellifera that were dark could be from C lineage Carnica bees, as they are also darker colored, unlike C lineage Ligustica bees, which are yellow? Well, just well, I think we answered
Similar the question.
Oh, I missed the question from Roger Patterson. What about coloring drones? Is that likely to be unreliable too? Well, I think so, because we did some drones that were quite yellow and they were not pure C lineage. But in this study, it was more worker bees.
Was what only workers, yes.
And from
That's a good question, though, you know, for the practical beekeeper. I know that Roger, you know, full-on selection, you know, bee breeder, and yeah, like with the drones, which are only heterozygous, they're all thinking, do they have a tool and how can they keep using color? Like full on, I can feel their brains working. Mine is too. So I think the question, so Dora, you say in your study that was all workers, no drones so far?
No, the one that Alice was telling, it is for other study, right? The drones that we genotype, but for this study, I'm almost pretty sure that was only workers.
yes,
okay.
it was only workers.
But there still is a chance, Roger. We can keep selecting for color on drones. Okay, we'll see.
And then there is a question from Matthew. I noticed that the maximum introgression seen was 0.6. Do you think these results are also applicable to pure CBs that might be in the UK population?
Well, if a pure C lineage beats yellow, right? Well, we don't know because we don't have that kind of bees. But I know.
Well,
I don't know one with 0.6, the one with 0.6, it has two yellow bands. But after we have another one with 0.5 that has completely black. But I don't know if we have like 1B that is 0.9, for instance, almost pure C lineage, if it's related with color or not, because we don't have that in our samples.
Well, I think if a B is a pure C, it should be 1. Q should be equal to 1. I mean, if it's pure. If you bring a Ligustica bee from Italy, it should be around 1. But after a few generations in a foreign area, like in the UK, the offspring will be whatever, right? Depending on the crosses. So I really don't know exactly what Matthew, what is his point? Because if it's pure C, it should be not 0.6, but 1 or 0, depending on the, right?
Yeah, I wouldn't figure that out. And although we don't know, like with the dark bee, we knew or we assumed that there's no yellow in dark bees, but with Carnica, we know that there are Carnica with yellow, like they are in the north, close to Italy and so on. But yeah, I don't understand the question either. Maybe Matthew can, but for Carnica is not, maybe we can, you know, like detail it a little more and we come back to it later, okay?
Now there is a comment from Vincent about the consequences of these results on conservation programs. When beekeepers are not able to get routine genetic monitoring, so they might think they do well, but they could increase pollution in their population. Yeah, so definitely, Vincent, it's not a good idea to use color. In order to help them conserving native bees, I would like to know what is the risk of a black bee not being Apis mellifera or an orange bee being an Apis mellifera. It should be probably meant linguistic, I think.
Well, the risk if it is a black bee but is not a mellifera is that you are promoting the genetic pollution because you keep the colony. If it's an orange bee and if you eliminate from the colony, from the population, you are eliminating diversity that can be important for the future of your population. I don't know if that is a question.
I think he wanted to have some numbers, you know, hard numbers, but I think it depends the population. It's probably different in the UK than in Ireland than on the Iberian Peninsula. It's like, like always, depending on the situation of the population, right?
Yes.
Yes, so
exactly.
girls, I think
you need the context. Yeah, we need the context to make better decisions, definitely.
I think we need to stop for the moment. Sorry, Alice. I think we need to go to Carlos's talk. Like everybody has sent questions, we keep the questions and we take our time to read it and maybe
Let me just finish with this comment from Niels.
Yes.
He says, thank you for your work. I made serious mistakes exterminating valuable colonies some 30 years ago based on yellow. Maybe not, Niels.
We all did.
Yeah.
Still do. We don't, you cannot sell yellow black bees. Right. Cannot do that either.
Well, maybe we can then take a look at these questions after Carlos' presentation, if we have time to answer these questions. Okay.
That's good. So Dora, we cut our videos and we give the floor to Alice and Carlos. Okay, and see you later.
Okay. Carlos Yadro is from Cuba, where he worked and he still works as an affiliation with a bee research institute in Havana. And he did his master's thesis on the genetics of Cuban populations using microsatellites and the mitochondrial DNA markers. And he has been also working with the honeybee diseases in Cuba. And about 1 year ago, he joined my lab. He came to work in a project, in Be Happy project. This Be Happy project is about the bees from the Azores that are very interesting because they are isolated from the mainland and from importation, and they have some islands have varroa, some islands don't have varroa. So this Be Happy project will finish soon. And meanwhile, Carlos earned a PhD scholarship from the Portuguese Science Foundation, and he will start doing his PhD in 1 month in looking at the genetics or the genomics, because he's going to use whole genomes to study the patterns and evolutionary processes in several species of the O lineage and A lineage from the Mediterranean area. This is a PhD that will be done in the context of a project that we have going on now called MedIVs. Carlos has a long experience with bees and is also passionate with bees. Today is going to present results of a software that we developed that is called Deep Wings, and this software is used for identification of bees using wing geometrics, morphometics, and the software was also developed in the framework of the BAPI project. And Carlos has been testing the software in samples originating from different places, mainly in Europe. Well, I give you the floor, Carlos. Good luck. You have to unmute your microphone.
Okay, now can you see the presentation?
Yes, it works.
Yeah,
well.
okay. Thanks everybody, and Happy New Year, of course, for all of you. Yes, as Professor Alice was commenting, this is a software developed in the framework of Be Happy, and it's intended to provide a tool for the beekeepers to select Apis mellifera through the identification of the wings using geometric morphometry. I'm sure that all of you have heard from, or many of you have heard from this program, because Professor Alice has been asking for wings and your help for this. Okay, so just a quick description of the program. The program is based in artificial intelligence. It's fully automatic, and it's composed of 3 modules. First, there is a pre-processing of the image, where the image is loaded, and it's made some modifications on the image to enhance the probability of the system to see the venation pattern. Then the wing is trimmed out of the image and standardized to a size, and then it goes to the landmark detection module, where the landmarks are set and extracted from the image. This module was trained with 5,770 right wings from Apis mellifera iberiensis and Apis mellifera ligustica. And in this module, in previous studies, we have had like 94% of average accuracy. And then once the landmarks are get from the image, it goes to the classification module that was trained with almost 500 right wings from 5 subspecies from rootnet collection: Apis mellifera carnica, ligustica, caucasia, and then Apis mellifera iberiensis and mellifera, of course. And then the classification is done. And in this one, we have got like 94% of average accuracy. I'm going to show you the program, because I'm sure this is the most interesting part of the talk. Okay. I don't know if it's running. Okay, so this is the home page of the program. You can see you have the 5 subspecies classifier, because there is 2 modes for 5 and 24 subspecies. It's only clicking the button and you can switch between the 24 and the 5 subspecies classifier. We're going to keep in the 5 subspecies for now. I think it's the most interesting part for us right now. And then is only drag and drop the wings, just like that. You drop the wings and the program starts to load the wings. And then processing, and then you get the results. You can have the lineage classification and its probability, and the same for the subspecies. And then you have cubital index and Hantel index and discoidal shift angle. They are also provided by the program. We have the cases like this for wing 8, where the program was not able to set all the 19 landmarks. So for the program, this is a fail in the classification. It will not show any result for this kind of wing. This is the first 94% that I was talking. And then, of course, you have the image of the wings with the landmarks already set, and to the right you have the coordinates of each one of the landmarks. We can download this table with the landmarks, coordinates, and the wings. And then, of course, you can download the results. Just only click here. And you have the results, and you have the probability for lineage and subspecies in numerical format. Then here, this part is not so important. This means that if the classification was successful in terms of setting all the landmarks. Okay, so the program is very easy. You just go, to go back, you just go home and you are in the home page. Okay. So as Professor Alice was talking, we have been testing the program with different data sets from different countries across Europe. I think we have like 12 countries and 15 independent data sets. And here I would like to take a small moment to say thanks to all the colleagues and keepers and researchers that have helped us by sending the image of their wings, because it was really good to have this representation, and it was only possible because of their help. You can see the names of all of them there. I'm not going to spend time because it's a lot of people. Okay, and before proceed, I would like to say that these results don't represent the real situation in terms of composition of the population, because we have a very unbalanced number of samples. We have, for example, in Belgium, we have only 2 colonies and 77 wings sent by DLAN, or even, for example, in Israel, we have 30 colonies and almost 300 wings, but all of them comes from a single apiary. Then we have, for example, other places like Ireland, that Grace send their wings, and in this case we have like 53 colonies, and I think most of them are distributed across the country. So in this case, it might be a good approximation of the situation. The same happens here in Poland. The wings that Adam sent for us, they are in 2 north to south transects in the center and eastern part of the country, and then we have wings that comes from colonies, from specific colonies, and we have other cases like in Poland and Hungary, where the wings were collected from flowers in the field. Okay, so it's important to keep in mind that our results doesn't represent the situation of the countries. Okay, so using the program, we get this result for each one of the data set. Here I'm showing all the wings that were classified, and we have, for example, in Sweden and Switzerland, Wales, in Auvergne in France, and in these two apiaries in the center and the eastern part of Russia, we have a large proportion of Apis mellifera of black bees. Then we have other data sets like Hungary, Romania, and Croatia, then Poland, where the most common subspecies we found was Apis mellifera carnica. We have, for example, other cases like Turkey, where the most common was Apis mellifera caucasia. And we have here this case in Russia that comes from an unprotected population. It's only 2 wings coming from 2 colonies, and all of them were classified as caucasia with very high probabilities. And then we have other cases where the results are more mixed. We have, like, for example, here in Russia, close to Leningrad, mellifera is still the predominant, but there is a very large frequency of caucasia and ligustica. Something very similar happens in Israel, where in Israel there is a lot of importation of bees specific, especially from C lineage. And in Auvergne we have a very similar result with a very mixed population. Okay, this is only this is using all the wings that we classified. In this case, the success in landmark detection was a little lower, was around the 90%. Okay, in white in each one of the charts, you can see the proportion of wings that was not classified. But if we consider exclusively the wings with very high probability, and for this we set an arbitrary threshold of 0.9. So now I'm showing only the wings that were classified with probabilities between 0.9 and 1. And while there is some difference, the general picture is still the same. We have mostly all the wings mellifera in Sweden and Switzerland, in Wales and Ireland and Auvergne, and then in Croatia, Romania, and Hungary, and even in Poland we have mostly carnica. So the results are very similar. Of course, if the user wants to keep only the wings with very high probability, for sure, but we don't think it's going to make a very big difference in the results. Talking about the probabilities, we also found that also the probability, the average probability in the detection, it can be related to the most common subspecies in the data set. We have, for example, in Wales and Ireland and Belgium, Sweden, in Auvergne in France, where Apis mellifera was the most common subspecies. It also have the highest average probability in the detection. Then we have other regions in Avignon. Apis is still high, but Apis mellifera carnica is very, very close, and even ligustica is considerably high, the average probability. In Switzerland, it's almost the same between mellifera and carnica. And then in Poland, there is a very large proportion of carnica and ligustica, and carnica is really, really high, the average probability. In Russia, most of the wings classified as Apis mellifera were with very high probability, except in the case of the one that is unprotected in the south center of Russia. So I told you it was only 2 wings classified as caucasia. And then in Croatia and Hungary and Romania, while we still have some Apis mellifera, but most of the wings were classified with very, very, very high probability, almost close to 1, as Apis mellifera carnica. The same in Hungary. The data set here was a little bit smaller, but it was the same picture. And in Turkey, it happens almost the same. This case, Caucasia, which is the predominant one, is very, very high in terms of average probability. And in Israel, the results are more mixed. I think like representing a little bit the composition of the population. Mellifera is around 90%, and Carnica is a little bit higher, even around, I think it's 93. But the results are very consistent what we get from the probability to the qualitative classification of the wings. Then, as we were trying the program, we decided to compare our results with other programs and molecular data, and we wanted to show you the correspondence between cubital index and Hantel index as estimated by our programs and other programs. Of course, we know that these indexes are very used by the beekeepers and very trusted because of by them. So we have, for example, here the correspondence between the cubital index calculated by Deep Wings, our program, and CB Wings. While the R-square is not very high, the slope is almost 1. The same happens in the data set from Ireland when comparing cubital index in this case and Hantel index in this case. So, of course, when we are talking about this, we can expect some difference in the estimated values of the indexes. First than anything, I think CB Wings and Draw Wings, the user needs to set the landmarks. Deep Wings does that automatically. But of course, that some variation is expected, but still, as we can see, the variation is not so high that we cannot trust in the result of Deep Wings. Important to say that Deep Wings doesn't do the classification based on the index, but based in the 19 landmark. It's a more holistic approach, okay? But since we know the beekeepers trust a lot in the indexes, we wanted to show, and that's why the program even shows the values of these indexes. But when it comes to the classification using these indexes, the results are still very trustable. Okay, we have here the classification. This is that set from Ireland, I think, from Grace. Yes, the classification using the estimation of the indexes by drawing, and then doing the classification in morphoplot using this range provided by the program. And then we have the deep wing classification. As we can see by cubital index and Hantel index and the scoidal shift angle, most of the wings classified as Apis mellifera mellifera using draw wing morph plot, most of them were classified as Apis mellifera mellifera using deep wings. Okay, the same happens with Hantel index here and with the scoidal shift angle. Then when we use the three indexes together to classify the wings using draw wing and morph plot, in this case there are more wings that are not classified as Apis mellifera mellifera, but of course using the three indexes simultaneously with a more restrictive condition that in these cases where we are using one index each time. Okay, so the still we wanted to show so we can trust in the results. Okay, then as I was saying, we compared our results with molecular information that was available for some of the colonies. Okay, this is from Ireland, also from Grace, she shared with us, and each dot represents a wing, and the colors represent the subspecies, and we have the probability in the classification using the wings. And the bars represent the the average M lineage proportion as estimated by microsatellites. And we can see that in general, there is a very good correlation in this data. Whenever we have very high colonies, wings classified as Apis mellifera mellifera with very high probability, in most of the cases, we have M lineage proportions over 0.9. Of course, we have some cases like this colony F104 that the M lineage proportion is very low. We have this one, for example, but in general, I think we have a very good adjust between this data. Something very similar we found for some of the colonies from Russia. Okay, these are the colonies from the center, not the ones classified as Caucasia, the other ones. And in this case, all of the Apis mellifera proportion, in this case is the Apis mellifera proportion also estimated by microsatellites. It's in all the cases is over 0.9, which resembles the morphometric classification of the wing most of the cases. Then we have these two particular colonies. They are from the western part of Russia, close to Leningrad. That it's really interesting because, for example, in this colony we have almost all of the wings were classified as Carnica and Ligustica, and still we have a very high probability of being Apis mellifera. In this one, the next colony, we have kind of the other side of the coin, because we have a lot of wings classified as Apis mellifera with very high proportion, and the Apis mellifera proportion by microsatellites is close to 0.5. Okay, but even I think yesterday we were talking with the beekeeper who sent this wing, and he was saying that he had this kind of problems for more colonies. He was going to send us some wings that he have a very high probability of being Apis mellifera by microsatellites, and then when he goes to the wings, they tell a very different story. Okay, but also using SNPs assays, we can see similar results. Here we are seeing the same, and in this case the bars represents the Apis mellifera proportion as estimated by the SNPs using 91 SNPs. These are from the array containing immune SNPs in immunity genes that Professor Alice was talking before. And we can see, for example, in France, in Avignon, when we have very mixed colonies with different with wings classified as different subspecies. Okay, we have Apis mellifera proportions that are even below 0.5. Okay, in the other case, in Owesan, where most of the wings were classified as Apis mellifera with very high proportion, you had very high probability. The Apis mellifera proportion was considerably high. I mean, the lowest is this one with 0.8 of Apis mellifera proportion. Something very similar we found in Israel, okay, where as you were seeing here, the colonies are very mixed. I didn't like to say hybridized in this kind of things, but very mixed with wings classified very differently in terms of different subspecies, and the Apis mellifera proportion estimated by SNPs is very low. In these cases, we think that the molecular identification kind of agree with the wing morphometry of the colonies. And finally we have the case from Wales. This were sent by Dylan, and in this case it's pretty similar. We still have colonies that are very mixed in terms of wing composition, and we have very variable also M lineage proportion. This was done with a SNP array from our group. I think it was using around 65, 68 SNPs, and we can see almost the same colonies with very mixed composition of wings. They tend to have lower M lineage proportion. Of course, we have cases like this where we have a lot of wings classified as mellifera with very high probability, and still the M lineage proportion is around 0.7. But in general, we think that the program is and it could become a very useful tool for beekeepers when it comes to the conservation of Apis mellifera. I mean, in general, it will be useful for Apis mellifera subspecies identification, because it is able to recognize 24 subspecies. Problem is that with the 24 subspecies, the average accuracy is kind of lower. It's around 80 to Apis mellifera, and the rest of the subspecies that are more commonly introgressed into Apis mellifera populations. We think it works fine. Then it's going to be freely available for pretty soon, I think. We will make it available. That means that we are providing a tool for the beekeeping that we hope is going to be very useful. Then it's an automatic system, so it minimizes a lot the bias associated with the landmark setting, because in some other programs you have to set the landmark, and of course, even if the same person is doing it, it's really complicated to be consistent in the landmark setting. And also the program reduces a lot the time you need for processing winds and for getting the subspecies identification. I think that when I was working with this, I was able to run around 1,000 wings per day, working like full speed, but it's really fast, the program. Then it's an artificial intelligence based software, what makes that it's easy to retrain the program to increase the accuracy and the precision. So eventually, as we are getting more and more wings with genetic information that we are sure about the subspecies, we can set the landmarks in wings from this beast, and we can feed the system again. And then, of course, I think the handicap is that we need to be very careful when analyzing the results, because depending, of course, in the context and the situation in general of the place where the samples have been taken, the morphometric classification might not resemble the molecular classification. So yeah, this work is part of the Be Happy project, and I would like to say thanks all of you for your attention, and a lot of thanks for all the partners that have sent to us wings so we can analyze and process and test our program. That will be all.
Thank you, Carlos. There are already questions. One from Lars. Working with Norwegian wings okay, Lars works with Norwegian wings. Will your software be available for downloading?
Yes, Lars, it's not for download, it's online. So, but very soon, I think you can access and try your wings, but it's online. You don't need to download or install. That makes even easier for us to retrain the program and to update the program with these new wings.
Let me comment on this. We haven't published yet the results, and even the software, the part of the development of the software, and we need to publish first before we make it publicly available. But we have been sharing with lots of different people, so if you want to try, we can just send me an email and we can send you the link. Well, I think before there are some questions, but we have to be careful about wings with morphometrics, geometrics, morphometrics. this is, it's not, I think it's better than color because we are looking at more genes probably, but we see that when there is admixture, then we'll get lots of variation with the results of the wings. And you know, I think it's always better to use molecular markers if we want to really know what is the proportion of introgression of our colonies. This can help, and if we don't have money to run molecular analysis, maybe you can have this as a first level guidance, but always better to have molecular markers. That's my my experience and my take-home message. Now let's see.
I have one question, so you have time to read the others. You showed us those plots, you know, with the, I guess the molecular marker was the average from the colony, because you showed, like, you know, the wing results as individual dots.
Yes.
But below it was just a bar. So was that the colony?
It's the average of the colony.
Did you also make a comparison, you know, from like single individuals? I guess you have microsats from individuals. And
we have only that for the data set from whales, from Dylan, and it was not so good because in the data set there is a lot of admixture.
But how did you get the microsatellite data? You must have individual microsatellite data.
No, for the microsatellites we only have the average of the colony, and it was sent by the beekeepers from Russia. In the case of SNPs, we have the individual analysis for whales, and I think also from the data set from France and Israel, but I didn't do yet.
Oh, but I thought you showed a lot of, maybe I misread it. You showed a lot of, you know, the plots with the dots.
Yes, the plots
and the bars below. Maybe you can show them again.
The plots, yes, sure.
I'm sorry.
No, for sure.
I'm thinking, why didn't he do a 101, you know, individual wing morphometry like Alexa did in his one paper?
Oh,
that was very interesting.
Okay.
Well,
yes, those ones.
Like here, okay.
Those, okay.
Okay, the dots are the classification of each wing by our program.
Like one individual bee.
Yes, one individual, and then the bars is the average, in this case, n lineage proportion as estimated by microsatellites.
For this group of dots.
For this colony,
For the whole colony, like an average, like you must have individual data, like you cannot pool microsatellite data, like DNA.
Yeah, the thing is that in many cases we didn't have the
you received
the individual microsatellites information. In many cases,
sorry, you did receive the microsatellite data. You didn't run it yourself, what I understand.
No. We received the microsatellite information. Okay. We didn't do by ourselves.
Yeah, okay, that's why. Okay.
That's why.
Yeah. Now I understand. Because it would be interesting to do a individual-based comparison there.
Yes, for sure. I think we have for Dylan, but I think it's not. I don't know if I have here. I think no, but it's not the best case because there is a lot of admixture, and the graph is very dispersed. But in any case, I remember that, for example, the wings classified as Apis mellifera, they used to have like medium to high M lineage proportion.
In the microsats.
In SNPs, in the case of Dylan, it was SNPs. Okay. Yes.
Mm-hmm.
Yeah.
So divergent
yes.
results, like you, so you didn't get the same results for the microsats and the wings.
Yes,
Okay.
it was, Dylan, it was this one. So
Mm,
as you can see, the composition of the colonies was very variable, right? But in general, most of the wings that were classified as Apis mellifera, they were like in the right top of the chart when you have here in here the probability of classification by big wings, and here the lineage proportion. So most of them were in here. So
okay. Thank you for clarifying.
No, for sure. Let me stop this.
Well, let me tell a little bit more about the program. Is because we developed it recently, and then we wanted to test it, and this is what Carlos has been doing with wings from different origins. And because some people have molecular data and ask, do you want molecular data for those colonies? So now let's use the data and get a clue about the two types of markers, morphometics and the molecular. But yeah, it will be much stronger if we would run a systematic evaluation of the wings against the molecular data for the same individuals.
Yeah.
The same individuals. And Dylan did that as part of his PhD for some of the colonies. And I don't know if Dylan is here, but
no, he's
something now.
A moment, sorry.
But he's supposed to write a paper. But yeah, this was just to kind of test the program to see how it performs with wings from different geographical origins and different subspecies.
So I'm glad that Balzer was able to send you wings, and he has a question there.
Yes, let me see. There is a question, Balzer. Oh, Balzer, he asked, why do you not consider buckfast as a subspecies by itself? I don't know. This question seems to be out of place. I don't know if it was related with the Dora's talk.
No, I think with the wings, you probably didn't consider it as a own subspecies.
Do we have wings of buckfast?
No.
From Balzer, though.
Did Balzer send wings of buckfast? I don't know.
No, when he sent, he didn't say about being buckfast.
No,
as far as I remember.
Probably not, but he
just sent the wing.
Buckfast is an issue, so I think he's just interested how you handle it. Balzer, maybe you can clarify some more, whether buckfast should be considered as a known subspecies or as like a hybrid. That's kind of the question.
I'm not sure about how to answer to that in terms of the program, because we train the program only with the Rudner's wings. So because this is not included.
The program works okay, but can, as Carlos said, can be improved. And this is the beauty of artificial intelligence, is that you can train. And the one problem is that we didn't have many wings from Rudner's collection. Only
I think in total it was like 2,000 in total.
Yeah, but like per colony, just
no,
from
per colony was only a few ones. Yeah,
a few, but especially not many colonies. So I'm not sure. These are the wings that have been used by many people around the world, that have been provided by the institute. I forgot the name. And it would work much better, if we would have a collection more representative of the range of each subspecies. And of course, then there is also the question whether we should use a collection that was collected many years ago. Do they still represent the present variation? Because meanwhile, the bees have been evolved, right? So, well,
That's
this is
always-
what it is.
And what's first, the chicken or the egg?
Yeah. Well, now there is a comment from Joe. If I sent a sample of bees from 5 colonies with native appearance, 5 with mixed, and 5 with buckfast appearance, I think the DNA analysis will bear that out.
I think this is for Dora, because before it was like Dora hypothetical comment or something.
Okay.
I'm not sure.
Okay, now it's from Witse. Have you also, apart from drawing and CD wing, compared the results obtained with the deep wings with Identify?
Yeah, at some point we were trying with the previous version of the program, and in terms of the more common subspecies in the data sets, the results were very consistent. I think we tried for Poland, and most of the wings were classified as Carnica and Mellifera by Identify. Yeah.
Just Identify is the
It's
same as
the last one from Adam.
It's the drawings. It's the same program, but modified.
Yeah.
But it should be okay, because you also presented the comparisons with Identify or with drawings.
With drawings.
With drawings, yeah.
With drawings.
But it's the same. A question from I don't know how to pronounce the name. Wout van der. Maybe you can pronounce it better, Gabriel.
I don't know. I would say Wout van der Verft. Maybe that's totally nice.
It sounds very professional. What will be the requirements to upload?
The requirements, I think.
Oh, the requirements to upload wing photos with regards to size and DPIs.
Okay. In terms of size, you can load in the program 10 wings or a total of 25 megabytes. So it's okay, because normally the wings that we have when we load, it's around 12, 15 megabytes, 10 wings in total. And then about the dots per inch, that's an issue that we are still needing to solve. Because, for example, we received some wings from Per Thunam that he sent by mail, and we tried to scan with, I think it was 600 dots per inch, and the quality was not good enough for the program. So we have to take photos with a microscope and a camera. So I cannot tell you right now the number of DPI that we need, because most of the wings that we have received, they were already photographed. But I can say, like, for example, taking photos with a good cell phone even, or trying to scan is not good enough for the program.
Okay. And a question from Megan Riley.
Wing asymmetry.
Okay, yeah. If you use a mix of, it's about using right and left wings. Would it make this less accurate?
Yes, for sure. The program was trained exclusively with right wings. So if you try left wings, the results are not gonna be good. For sure. Even there is very big difference in the classifications. So yes, only right wings.
Yes, because it was trained. Did we? I think you tried.
Yes, I tried.
With right and left wings, and the results were different.
Were very different.
Have a bee right-winged Mellifera and left-wing Carnica or something, or I don't know.
No, but I think the venation pattern might have some difference. I don't know for sure, but the results were very different.
Like this one, different subspecies, different, or
no difference in very different. The classification.
I mean, the probabilities were different.
Even in the classification, there was some differences.
Yes,
in the
because,
subspecies classification.
Oh, this is great, guys.
yes, like the program, it was trained. This is artificial intelligence. It's based on training. It was trained with right wings. To be able to use both, it would have to have been trained with both right and left wings.
And you should be able to, like, you know, mirror the left wing. But this means, like, if I can, you know,
It's curious because the programs does that. You know, if you put the right wing in a different position, the program will take it to the position of the references.
Yeah.
So it's not about flipping the left wing.
Yeah, well, you know, when you only have left wing, but that means that beekeepers, when they use that, consequently, they will be selecting for proper right wings, but then the left wing. So they might have bees like this at the end. My imagination is running wild just at the moment.
This is the landmarks, the position of the landmarks.
Yeah.
A question from Matis. Thanks for the talk. Is drawing identify? Yes, it's the same. What were the conclusions? Identify is very strange to me and cost a lot of time.
Yes, for sure. With Identify, you take a lot of time to set the landmarks, and then when you get the results, the probabilities that Identifies gives back are kind of really, I don't know, really strange. You have wings with 0.00007 of being a subspecies, and that's in the best case sometimes. So yeah, Identify, I'm not sure about the perform itself of Identify.
And a related question from Wout van der Berg. I also wonder if the way of analyzing is close to Identify.
Wow. For that part, I'm not completely sure because I was not involved in the developing of this program. It was a colleague from us who works with artificial intelligence. So I cannot answer that question right now for sure.
Yeah, I think it uses a different approach. Yeah, because it's based in artificial intelligence. Anyway, another difference with Identify is that you have to set up the landmarks yourself.
Yes.
Whereas here is automatic. It's one advantage because the same person, if places the landmarks in different days, they may be in different positions. So this person variation is removed from doing the automatic location of the landmarks.
Yes, here with deep wings, we can be sure that it's consistent.
Consistent, yeah. And a comment from Vincent Duar. Time to offer such solutions, so much needed. Our amazing mixed population in Avignon. We will train your program when it will be public. Okay. Yes, population Avignon. It's just from one apiary, and yes, it's but it's known in France. There is lots of admixture going on. And related question from a question from Enk. Enk, do you compare deep wings with Identify from dolphins?
I think we already talked about this.
And from Roger Patterson, what is the long-term future of the program? How long will it be supported and updated?
I think maybe it's better to answer this one.
Well, it will be available for many years, I hope.
Yeah.
And if we get students to do more training with more wings, to train for subspecies, it can be improved. But one problem is that what is it's the ground truth, because for now we assume that the Rutner's collection is the best representation of the subspecies, but maybe this is questionable, and we have problems with it. Maybe if we would run some molecular analysis and take this as the new representation of the species, then we could do the analysis of the wings and assume that they represent the ground truth. So we can still do a lot to make it better, the program. But the version that we have now, it will be available for many years.
I have like a question for this Rutner collections. Has it ever been analyzed genetically? Like, do we have like the genetic, like whole genome sequences, and for this Rutner collection that everybody?
I don't know.
It is something interesting to see whether they have been. Like, one question is, has this reference population been pure at that point still? And what diversity of the populations, of the original populations, do they represent, right? Those are the two main questions.
Well, but subspecies were defined by Rutner based in morphometics.
Well, no, he started doing that. It has all been confirmed with the genetics after, with the mitochondrial lineages and so on. But now that everybody's using the Rutner wings again, it would be interesting. Now we have the tools, the genetic tools, to verify that. Now that you yourself are putting it in question, we don't know.
Mm-hmm. Are you talking about the collection that he collected?
Yes, the wings you used, the Rutner wings,
It will be interesting. It's a pity that maybe Marina Meissner is not here, but she could answer that question. But I think it will be a nice project to run molecular analysis on the bees of the Rutner's collection.
That would be interesting to verify, so we know for sure that those are good collections.
And to see how much variation they capture, that collection.
Exactly.
A question from Witze. Could this right-wing, left-wing issue be an effect of hybridization?
Well, I'm not sure about that. So far I never read anything about that issue. Then there is another comment saying that from John Durkash, that he said that he read an American study that the difference between right and left wing could be from developmental stress. So maybe I don't know. What I have heard about developmental stress is more about the size and the perform of the bees, but not like difference between the wings. But I don't know. Maybe
there is wing asymmetry. I know about that. And nutrition can affect wing symmetry. And there are some works, I think, published by Tiago Francois from Brazil, dealing with that issue. So there is asymmetry, and it's influenced by environment. But also by genetics.
I remember that paper from Middle America somewhere. You might, it's probably the same, but it was related to hybridization, I thought.
I think that both components, genetic and environmental, will influence. A question from Barbara. This is very interesting. Which platform will it run on? Asking for a mainly Mac user, or might it be run from a browser? You can answer this question.
Yes, for sure. I mean, so far I have only tried in Windows and Chrome. But I think that when Grace was testing the program, she was having problems because she was running on Mac and I think Safari. But and the developer of the program made some fixings. It's the program to run on Mac, but I don't know if Grace managed to run the program. She never gave us feedback about that. But in theory, it should work in Mac.
There is another comment from Sven. Would it be possible to upload a picture with multiple wings? I think that at least 30 wings are needed for a reliable result.
No, it's not possible to run an image with several wings together. You have to crop and take individual wings.
Yes, yeah, it was trained with individual wings.
Yeah.
And a question from Machis. How reliable is this program for beekeepers who want to keep native bees but can't do DNA? Is this a good solution?
I think it was the purpose of. I think this was the purpose of the program, to provide the beekeepers with a tool. So I think yes. I mean, many beekeepers, they rely only in cubital and handle index to do the classification. So using all the 19 landmarks is a more holistic approach to the classification of the wings. So I think that is a good solution in terms of all the landmarks that you are using. That of course, using the 19 landmarks, you are capturing a wider picture of the genetic variation that only using the traditional indexes. And also it's a fast method. So yeah, I think it's a good solution, and the possibility that we can retrain the program eventually, it will make it even better.
Well, it's a good solution, but it does not replace the molecular analysis.
No, for sure, but yeah, molecular is expensive, and not everybody has access to.
And a question from Lars. For me to use the program, what will be the crucial in the preparation of the wings? Yeah, this is
I think you only have to be careful to have complete wings, because we have had cases that the wings are kind of broken, like in the proximal part of the wing, and the program is not able to recognize. So yes, trying to have the wings complete, try not to be fold or bended, that kind of things. But in general, the program, and that you can see the divination pattern well.
Okay, and I think there is a last question. I hope I didn't miss any. A last question from Gavin Ramsey. Do you know how many genes control the wing traits you are scoring? And then he comments, my impression is that there are rather few, because it's not difficult to find hybridized bees with wings that look right for your chosen subspecies. Many times in the past, people have overemphasized the use of wing traits in the breeding of honeybees. From my point of view, they can only help you exclude hybrids from breeding, not confirming taxonomic identity at all. It's a good
yeah, well, I'm not sure about the genes controlling the wing traits, for sure. And of course, when you are using, let's say, whole morphometry of the bees, you probably are going to get better results than using exclusively the wings. Because even then, of course, I think Dylan was talking about this with us. If you select your bees based only in the wings for a very long time, eventually this selection pressure is gonna mean, it means that eventually you are going to have the wings that you are selecting, but that doesn't mean that you don't have other subspecies hybridizing and leaking genetic variation into your population. So yes, for sure, it's not the perfect tool, but as we were saying before, it's a good choice when you don't have access to molecular information.
Let me further comment on this, because I think what struck me in these results that we have so far is that, for example, for Ireland, where there is no history of introgression, the program was very consistent in identifying most of the wings above 0.9 probability.
Yes.
I think the problem, we start to have problems when there is hybridization. Then we get all kinds of probabilities. There is lots of variation in terms of the probabilities. So I think it's the other way around. I think this works better in areas where there is no introgression, because we saw that for Ireland and we saw that for Russia, right? For Russia, they were really clean. I mean, the results, they just...
In some places, it was really clean. Even in our some were really clean.
Mm-hmm. And we didn't show results for invariances, but for example, for the colonies sent by Dylan from conservation area, also most of the wings were above 0.9.
Yes.
Mm-hmm.
But that just shows again, you know, the difference between, it's like two different questions with the DNA and the morphometry. And I think what confuses a lot the beekeepers with the DNA. And I also wanted to put that as a last question or a comment. With the DNA, you also get, you know, these probabilities between 1 or 2 or 3 groups. You know, it always adds up to 100% probability. So, and we always put a threshold and we say, okay, above 95%, it's a pure mellifera. We accept this as a pure mellifera, and the others are identified as hybrids. With the morphometrics, it's not a tool to identify hybrids, really. It's more a tool to identify pure subspecies, right? In a pure environment, identify, is it a pure mellifera or a pure carnica? But it's, as you say, Alice, once you come in a situation where you have all hybrids, it doesn't work anymore. And I was noticing that with your, you know, with the bars, that I was very focused on, you know, the plots with the dots and the bars. It was fascinating me a lot. But I noticed that you never identified a measured dot as hybrid. You said it's a carnica 60%. Like with DNA, you would say it's a hybrid, you know? How can it be a carnica 60%? That's nothing. That's all over the place. How can you even identify or like assign it to a group with that probability? So it's like apples and pears, right?
Yes, for sure, for sure. I mean, when we talk like internal, yes, we have set this 90% threshold in the classification.
Okay.
But of course, then, and of course, you know, it's a way of seeing that below that threshold, wings can be considered as hybrid. But I don't know, I didn't like to use the words hybrid or pure breeds when it comes to morphometry. Some people doesn't like. Of course, that's the approximation and the easiest way to interpret these kind of results. But yeah, some people doesn't like.
Well,
let me also make a comment, because when we do the analysis with the molecular data, we run, for example, a structure or a mixture analysis. What we get is a membership proportion. It's not the same as a probability.
Yeah,
they are two different things.
Yes, true.
So that's why we can tell with the membership proportion that this bee has this proportion of these of being variances, like let's say, for example, 60% of being a variance plus 40% of the genome comes from a different origin. You know, it's different from a probability.
Well, the structure, it says in the manual, it's a probability of belonging to, you know, group A or B or 3. So yeah, well, that's, I think for the beekeepers, it's kind of semantic.
But you know, it's a partitioning of the genome. And in a study that we run and we published, I think it was in the paper in Scientific Reports with a SNP assay for Apis mellifera mellifera, where we did crosses. Actually, it was Per Krieger that did in Denmark between mellifera mellifera and I guess Carnica. And we analyzed the F1s and we got really clean and beautiful data. It was not 0.5, but it was like 0.55
Yeah.
0.6, 0.4, very close to 0.5.
Yeah.
Very, very
40 and 60% generally.
So the part of the genome, it's the partitioning of the genome. Part of the genome comes from that subspecies, and the other part comes from the other. So this is probability. So it's not comparable. It just gives you a probability of being. For example, when we analyze with the 24 subspecies, doesn't work.
Yeah.
It doesn't work so well as with the 5 subspecies.
Yeah. I see.
Well, there are two, I think, well, we can, oh my God, there are so many questions, but I think.
Hang on, no, the Gavin and Megan have been answered. So I think, Harman, hi Harman.
Harman,
Oh.
it is not both ways, the wings should be correct and also the DNA.
Because I think I suppressed Megan, who was not, hang on. Megan asked before, sorry, to add an extra question. In order to get an estimate of the purity of a whole colony, how many individual bees would you suggest using? No, that wasn't the question. Sorry, Megan.
Oh, no, yeah. Well, of course, the more wings you have, the better. But we have been using around a minimum of 10 wings per colony. I think that's a good number. Then, of course, if you can reach 20 or 30, for sure, it's better. But I think that 10 can give you an approximate idea of what you have.
And from Harman, is it not both ways the wings should be correct and also the DNA?
Yes.
I think it's asking about the difference, right, between morphometics. Well, the thing is that with the wing morphometry, you are only taking a small picture of the whole genetic variation. You are only seeing the results of the genes involved in the morphometry of the wings. Then when you go through SNPs or microsatellites, you are seeing a picture taken from another side of the problem. That's the thing. Of course, the ideal will be that they go together, but doesn't have to be that way.
Yes, it's like what I said about color. If you just have a few genes controlling one trait, and the wings, I don't know how many genes control the wing shape, but we are just looking at a very small portion of the genome. Whereas when we run SNPs or microsats, we may sample across the entire genome. For example, for SNPs, when we design the assays, we make sure that we have SNPs for the 16 chromosomes. So we sample the entire genome. And besides, the wing shape is also influenced by environment, whereas when we look at the SNPs or microsats, we are removing the influence of environment. So they don't have to be coincident, and they are not. Yeah.
So
and
one last from John.
There are localized pure island populations in Scotland. Yes, I think we analyzed wing morphometry there gives excellent AMN results as they are closed populations. Yeah, I don't know what island is talking about.
You're talking about
maybe Colonsay or
Colonsay,
something.
yeah. We don't have wings from Colonsay.
No, we don't have.
I bet that they would get results like we get for Ireland, probably like clean. But it will be nice to analyze wings from Colonsay. Well, I think that's it. It has been a long.
That was very nice and very, very interesting. I'm sure that many people will go and have a look at those talks and the Q&A as well. So thank you very much to all of you. Those are, questions that are very pressing to all the beekeepers that don't have access to DNA, and the morphometry issue is always rediscussed very buoyant, like heavily. So that was very nice. Thank you all for taking the time. Yes, Barbara Keating says also brilliant night. Thank you so much. So yeah, thanks. Applause.
Thank you.
Thanks, everybody.
Thank you, everyone.
I would like to say goodbye to everybody, and please join us again next week with Sonja Leidenberger from Sweden. She will give us an insight about the activities there in the scientific field. That will be very interesting as well. And we give you some minutes to exit the conference, and hope to see you again next week. Thank you. Bye-bye.